Cytology, the study of cells. Cells were first discovered in 1665 by Robert Hooke, who viewed dried cork under a light microscope. The cell theory has three components. 1. Cells are the building blocks of life. 2. Cells are produced by the division of pre-existing cells. And 3. Cells are the smallest structural units that perform all vital life functions. Cytology is the study of the structure and the function of cells. The fluid surrounding cells is called extracellular fluid. The cell membrane separates the cytoplasm from the extracellular fluid. The cytoplasm of a cell contains cytosol, fluid, and organelles. This is a quick help sheet for the components of the cell. The cell membrane is called the phospholipid bilayer because it has two distinct layers, hydrophilic heads and hydrophobic tails. There are two general types of membrane proteins: peripheral proteins attached to the inner or outer membrane surface, and integral proteins embedded in the membrane. Some integral proteins have channels that control the passageway of materials. The cell membrane provides physical barriers, regulation of exchange, sensitivity, and structural support for the entire cell. It should be noted that the membrane structure, however, is fluid. Cholesterol is used to help anchor structure together. Membrane permeability determines barrier effectiveness. Cells are selectively permeable. The permeability of the cell depends on passive and active processes. Passive processes include diffusion, osmosis, and facilitated diffusion. Active processes include active transport and endocytosis. Diffusion is the net movement from an area where its concentration is relatively high to an area where concentration is relatively low. The difference between high and low concentrations is the concentration gradient. Cell membranes are permeable to water molecules. The transfer of water molecules from high to low concentration is called osmosis. Compounds such as glucose and amino acids can be transported across the membrane by special carrier proteins. This is known as facilitated diffusion. The molecule to be transported first binds to a receptor site on an integral membrane protein. All active membrane processes require energy, ATP, to allow the cell to transport substances against concentration gradients observable by active transport and endocytosis. Active transport requires specific carrier proteins but does not depend on a concentration gradient. Ion pumps move a specific cation or anion in one direction into or out of the cell. Endocytosis, also known as bulk transport, involves three processes: pinocytosis, phagocytosis, and receptor-mediated endocytosis. What is the difference between pinocytosis and phagocytosis? Pino is to drink and phago is to eat. Receptor-mediated endocytosis is similar to pinocytosis, but vesicles that enter the cell membrane are coated by ligands bound to receptors on the membrane surface. Small projections of the plasmalemma are termed microvilli, which help by absorbing material from the extracellular fluid, such as the cells of the small intestine and the kidneys. The general term for material inside the cell is cytoplasm. Cytoplasm includes cytosol and organelles. Cellular organelles can be divided into two broad categories: 1. Non-membrane bound organelles, and 2. Membrane bound organelles. The internal protein framework that gives the cytoplasm strength and flexibility is the cytoskeleton. There are four major components: microfilaments, intermediate filaments, thick filaments, and microtubules. Slender strands composed primarily of the protein actin are termed microfilaments. Microfilaments have two major functions. 1. They help by anchoring the cytoskeleton to the plasmalemma proteins, and 2. They help interact with microfilaments or myosin. Intermediate filaments provide strength, stabilize the positions of organelles, and transport materials within the cytoplasm. Intermediate filaments are specialized, and they're also called neurofilaments. They are found in neurons where they provide support within axons. Thick filaments composed of myosin protein subunits are abundant in muscle cells where they interact with actin filaments to produce powerful contractions. Microtubules. All cells possess hollow tubes called microtubules made from tubulin. Microtubules have the following functions. They provide cell strength and rigidity, attach to organelles, form spindle apparatus, and form structural components such as centrioles, cilia, and flagella. Centrioles organize microtubules in the spindle to move chromosomes during cell division. Cilia propel fluids or solids across the cell surface. Flagella propels cells through the fluid. The centrosome is the region of cytoplasm surrounding the pair of centrioles. It directs the organization of the microtubules of the cytoskeleton. Ribosomes are small, dense structures that cannot be seen with the light microscope. There are free ribosomes and fixed ribosomes that help manufacture proteins. Mitochondria contain an unusual double membrane, an outer membrane and an inner membrane containing cristae. Cristae increase the surface area or the matrix of the mitochondria, similar to a bookshelf. The nucleus is the control center of the cell. A nuclear envelope surrounds the nucleus and separates it from the cytosol. The nucleoplasm also contains a network of fine filaments called the nuclear matrix that provides structural support and may be involved in the regulation of genetic activity. Each chromosome contains DNA strands bound to special proteins called histones. At intervals, the DNA strands wind around the histones, forming a complex called a nucleosome. The nucleus also contains chromatin, which is loosely coiled filaments. The endoplasmic reticulum is a network of intracellular membranes that form hollow tubes, flattened sheets, and rounded chambers called cisternae. The endoplasmic reticulum has four functions. 1. To synthesize carbs, steroids, and lipids. 2. To store molecules or substances absorbed from the cytosol. 3. To transport substances within the cell. 4. To detoxify cellular toxins. There are two divisions of the ER, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The Golgi apparatus consists of flattened membrane discs called cisternae. The major function of the Golgi apparatus includes the synthesis and the packaging of secretion, packaging of special enzymes for use in the cytosol, and the renewal or modification of the plasmalemma. Lysosomes are vesicles filled with digestive enzymes formed by the rough ER and then sorted by the Golgi apparatus. Primary lysosomes contain inactive enzymes which rapidly destroy the proteins and organelles of the cell in a process called autolysis. Peroxisome enzymes are formed by free ribosomes within the cytoplasm. Peroxisomes absorb and break down fatty acids and other organic compounds. Large areas of opposing plasmalemmae may be interconnected by transmembrane proteins called cell adhesion molecules, or CAMs. There are three major types of cell junctions. 1. Tight junction, 2. Communicating junctions, and 3. Anchoring junctions. Tight junctions involve interlocking membrane proteins. Communicating junctions are also called gap junctions. They involve two cells held together by membrane proteins called connexons. 3. Anchoring junctions provide mechanical linkages between two adjacent cells at their lateral or basal surfaces. The life cycle of a cell. Cell division of one cell into two daughter cells requires mitosis. The steps of mitosis include interphase, G1, S, and G2 stages, then mitosis followed by an indefinite period of time. G1 involves normal cell functions plus cell growth, duplication of organelles, and protein synthesis. The S stage involves DNA replication, synthesis, or histone formation, and G2 involves protein synthesis. In DNA replication, the original paired strands unwind, and DNA polymerase begins attaching complementary DNA nucleotides along each strand. This process produces two identical copies of the original DNA molecule. Adenine, as a nucleotide base, is always paired with thymine, just as guanine is always paired with cytosine in DNA. These are the 4 stages of mitosis: prophase, metaphase, anaphase, and telophase.